Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 29th January 2021. As we are all familiar that we are into the revision mode and we are revising every chapter with the first to start with. I had given you the notes, revision notes, giving focus to the areas that are important in each chapter. Second, we have done assertion and reasoning type questions. Third, we have done test paper 1, and now fourth, test paper 2. So this gives a fair idea about where to focus, and also it gives you what is your strength and weakness. So in this session we are doing all important chapter number 12, biotechnology and its applications. So here we go. 1. Gene encoding hirudin is synthesized to A. Decrease stress, B. To increase sugar level in blood, C. Prevent blood clotting, D. To prevent high blood pressure. Question 2. Plasmid used to produce humulin is A. PAC, B. YAC, C. PBR322, D. phase. Question 3. Dolly sheep is made by implanting oocyte at what stage? A. Secondary oocyte, primary oocyte, morula, D. blastocyst. Fourth, gene for controlling production of insulin is beta galactidase. It can be used for A. PCR, B. ladder, C. blue-white screening, D. gel electrophoresis. Question 5. Most industrialized nations are rich financially but poor in A. population, B. biodiversity and traditional knowledge, C. traditional knowledge and land requirement, D. health and manpower. Question 6. ADA deficiency leads to enzyme isolation, immune dysfunction, C. immune response, D. DNA hybridization. 7. Cry genes introduced in plant will not affect against Coleoptera, Hymenoptera, Leptoptera and rodents. Question 8. Descriptive type from now onwards. Name the vector used for transferring ADA DNA into the recipient cells in humans. Name the recipient cells. Question 9. Which transgenic animal is used for testing the safety of polio vaccine? Question 10. What is golden rice? Question 11. Explain why children eating golden rice are unlikely to suffer from night blindness. Question 12. What was the specialty of the milk produced by transgenic cow Roji? Question 13. Case-based type. These are the new type of questions that are being introduced by CBSE for your batch. So you need to be careful about this. Arvind's mother has developed diabetes. Doctor suggests her to take insulin injections. But his mother declines as she presumes injections are prepared by slaughtering of animals. How will you solve his mother's problem with your knowledge of biotechnology? Question 14 again, case study type. Inspector Dubé could find only a few hair strands from the crime scene. He wants to proceed for DNA fingerprinting, but the amount of DNA is very less. In your opinion, what could be the solution to this problem? Write the basic steps in this technique. 15. Diagrammatically represent the experimental steps in cloning and expressing a human gene into a bacterium like E. coli. Now take a paper, start writing the answers. It may take say 30 to 40 minutes, and once you finish, come to the answer session. Compare your answers with the answers that are provided here. So here we go. Question 1. It should be C because hirudin prevent blood clotting. Hirudin is an anti-clotting agent that prevents blood clotting. It is obtained from secretion of leeches. The gene encoding hirudin is synthesized to prevent blood clotting during surgery. Next, question 2C as expected, PBR322, plasmid or extranuclear genetic materials present in bacterial, Humulin is produced by using plasmid PBR322. It's an E. coli plasmid vector containing the ORI, origin of replication. Question 3D, obviously blastocyst. It's Dolly, 5th July 1996 to 14th February 2003, was a female domestic sheep and the first mammal cloned from an adult somatic cell using the process of nuclear transfer. Dolly had three mothers, one provided the egg, another the DNA, and the third carried the cloned embryo to term. She was created using the technique of somatic cell nuclear transfer. This is the technique, where the cell nucleus from an adult cell is transferred into an unfertilized oocyte, that is developing egg cell, that has had its cell nucleus removed. The hybrid cell is then stimulated to divide by an electric shock, and when it develops into a blastocyst, it is implanted into a surrogate mother. Dolly was the first clone produced from a cell taken from adult mammal. See, normally we say God creates everything. Now scientists, being human beings, they are creating the new animal. So it's the artificial creation. We are stepping inside through biotechnology. Question 4C, blue-white screening. The blue-white screen is a screening technique that allows for the rapid and convenient detection of recombinant bacteria in vector-based molecular cloning experiments. DNA of interest is ligated into a vector. The method is based on principle of alpha complementation of beta galactidase gene. This phenomena of alpha complementation was first demonstrated in work done by Agnes Ullmann in the laboratory of Francis Jacob and Jacques Monod, where the function of an inactive mutant beta galactidase with a deleted sequence was shown to be rescued by a fragment of beta galactidase, in which the same sequence, that alpha donor peptide, is still intact. Question 5th B. Biodiversity and traditional knowledge. Most industrialized nations are rich financially but poor in biodiversity and traditional knowledge. Biodiversity and traditional knowledge related to bioresources can be exploited to develop modern applications. 6th, immune dysfunction. B answer. Adenosine deaminase deficiency is an inherited condition that affects the immune system and typically leads to SCID, severe combined immunodeficiency. People with SID have a reduced or absent immune response that leaves them vulnerable to frequent bacterial, viral, and fungal infections. Most people affected by ADA develop symptoms of the condition before 6 months of age. Question 7D, rodents. Cry gene produce toxin protein that chokes the gut of insects in the presence of alkaline pH to cause death of insects. It is not effective against rodents. It is effective, very, very effective against all the insects but not rodents. Question 8. Retroviral vector, ADA, cDNA, lymphocytes. Question 9. Transgenic mice are being used to test the safety of polio vaccine before they are used on humans. Normally even COVID-19 also, it is tested first in animals, then volunteers, then on large scale human beings. Then only it is marketed openly. Question 10. Golden rice is a variety of rice, Varija sativa, biological name of rice, produced through genetic engineering to biosynthesize beta carotene, a precursor for vitamin A. In the edible parts of rice, it is intended to produce a fortified food to be grown and consumed in areas with a shortage of dietary vitamin A. Golden rice differs from its parental strain by the addition of 3 beta-carotene biosynthesis genes. The rice plant can naturally produce beta-carotene on its leaves where it is involved in photosynthesis. Question number 11. Mainly children become blind every year, 50% of whom die within a year of becoming blind. Nearly 9 million children die of malnutrition every year. Very unfortunate. Vitamin A deficiency severely affects their immune system. So thanks to golden rice has been engineered to contain the genes necessary to make up the biochemical pathway for pro-vitamin A production. Moreover, the genetic construct was designed to be expressed exclusively in the rice endosperm, that is, in the edible part of the seed. The intensity of the golden color is an indicator of the concentration of beta-carotene in the grain. Question 12. The first transgenic cow, Roji, produced human protein-enriched milk, 2.4 grams per liter. The milk contained the human alpha-lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Question 13. Insulin can also be synthesized in the laboratory with recombinant DNA. Values: empathy, awareness, and concern. Question 14. He can amplify DNA. Of course, the inspector got the hairs, only some little hairs, and the DNA available from the hairs will be little, and the amount of DNA that is available in the crime scene can be enhanced, can be amplified with the help of a technique called PCR technique. So in this polymerase chain reaction technique, 3 steps will be there: denaturation, annealing, and extension. What are the values that inspector is displaying? Critical thinking and awareness. Question 15, long answer type. DNA cloning is a method of producing multiple identical copies of specific template DNA. It involves the use of a vector to carry specific foreign DNA fragment into the host cell. The mechanism of cloning and transfer of the gene for growth hormone into E. coli is represented below. Like you have got to take a human being, that is the growth hormone DNA, and here you can see E. coli and the chromosomal DNA, that is plasmid, and plasmid is extracted out, and then this is cleaved plasmid. Now this is inserted, the fragment of DNA, that is we say gene of interest, is inserted in this, and this is with the help of an enzyme called ligase, this is joined, and you get that recombinant plasmid DNA. And now through transimmunation, this is to be enhanced. The numbers are to be increased because this has got a plasmid with a gene of interest. So the numbers, as we increase the E. coli numbers, automatically the gene of interest also increases. So every answer, long answer type answer particularly, please draw diagrams and substantiate your answer with the diagram. So this is how you must practice. Next session we will see one more paper.